Transcription is a process that involves the transcribing of genetic information from DNA to RNA. The transcribed DNA message is used to produce proteins. DNA is housed within the nucleus of our cells. It controls the cellular activity by coding for the production of enzymes and proteins. The information in DNA is not directly converted into proteins, but must first be copied into RNA. This ensures that the information contained within the DNA does not become tainted. There are three main steps in the process of transcription. First, the RNA polymerase binds to DNA. Then the RNA polymerase transcribes a single strand of DNA into a single-stranded RNA polymer called mRNA. RNA polymerase releases the mRNA polymer and detaches from the DNA. A single DNA sequence can be transcribed by many RNA polymerase molecules at once. As a result, proteins can be manufactured in large quantities. Once the mRNA polymer is created, it crosses the nuclear membrane into the cytoplasm of the cell. The molecule is then translated for protein construction. DNA consists of 4 nucleotide bases: adenine, guanine, cytosine, and thymine, A, G, C, and T, that are paired together, A with T and C with G, to give DNA its double helical shape. Nucleotide base sequence are the genetic code of instructions for protein synthesis. This is my own 4-codon DNA sequence. Translation begins when mRNA binds to a ribosome. The first transfer RNA occupies the P site of the ribosome. Another set of transfer RNAs with bound amino acids first enters the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called anticodon, and 3 nucleotides on the messenger RNA, called codons, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which no longer carries an amino acid, leaves the E site, and the next transfer RNA with the complementary anticodon enters the A site. Each time a new codon sequence moves to the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid is carried. It's transferred to the growing amino acid chain. As the ribosome proceeds down the mRNA, a stop codon is finally encountered. The ribosomal complex then falls apart and the protein is released in the cell. Each codon in mRNA codes for a specific amino acid. Each codon in mRNA codes for specific amino acids, and when amino acids come together, they build a protein. This is my final protein.